your postdocs and in your careers, whichever they might be, you should really think about. And one thing that we are trained to do while we are PhD students is to think critically about everything that surrounds us. So not only in terms of the science that we are doing, but also in terms of the system. And this is precisely what I could realize you were doing while discussing issues that we face in terms of publications. I don't know what else you were discussing before, but I could definitely understand that you were talking about problems with publications and high fees the journals charge, and how the decisions that some of these journals influence our careers. And they have editorial criteria. They decide which topics are hot, and if something gets published in Nature or Cell or Science or any of these big journals, then just because it was published there, it becomes hot. And that many times, as you were talking about, the science that is supporting these publications is not always that sound. And the hotter things are, sometimes the shakier the science underneath is. And this is something that, of course, is a concern. But I don't want to sound negative, because there was something that excited us and that made us all want to be scientists and do our PhDs. And despite all these issues and problems, we're all here. I hope you're all enjoying what you're doing in your PhDs and that you're learning a lot. And I think you should try to keep this excitement and this motivation, because life, whatever it might be, will always bring you challenges and difficulties along the way. And the point of maturing as a person and as a scientist is also to learn how to overcome those problems and issues. And the best thing you can do in your lives, whether you decide to stay in science or to go do something else, is to make your voices heard. And because I'm a scientist, or I've been a scientist for some time now, and I've seen the system from the inside as well, I actually wrote an article about some issues I think we face in science. Maybe I'll just show this manuscript briefly, then you can look it up. I believe you're seeing this now. It was published not so long ago, just a few months ago in EMBO Reports, and it was, I used the title, "The Courage to Change Science." So you can see it's not very long, so you can read about it, and you can see if you agree with some of these ideas or if you disagree. But the point was that indeed, I think we need to speak up, and we need to speak up against the things that we think need to be changed. And it's very difficult to do, because we are all involved and because we all depend on the system. And it's not so easy that we speak up against these decisions that these journals make, because of what you were also mentioning, because then you don't know if you will have your access to some of these journals blocked in case you need to publish, or in case you have something you want to publish there. And then, of course, this will affect your career. So what I think needs to happen is that the people that are in the top positions, that run institutes and that run organizations that are at a higher level, they need to really speak up against all these issues, and they need to do something. We can complain, but ultimately the people that will be able to do something and to change the system are the people that are running the show and that are really in charge. And so we can only try to put pressure on those people so that they can really change the system and change the way we are evaluated, and that it doesn't depend so much on numbers, at least on impact factors, which, as we know, can be very inflated in many different ways, but in other metrics that are actually more fair, and that show in the end the work that we do and how valuable it is. And it's also not, I'm also not a fan of now going and forcing unnatural collaborations between basic scientists and companies just to show that you will develop something that will become a company or that will become a product, because this is also not always what happens. I mean, we need basic science, whether it will lead to an application in 1 month or in 1 year or in 2 years. We need basic knowledge, and this can only be achieved with basic science and basic scientists, and this needs to be funded because it's an investment in the future. We have many examples of basic science that was funded and supported, and that at the time no one really knew what that was useful for. And then years or decades later, it became evident that, that discovery that was done years before was very important for a very practical application. So anyway, I'll stop talking about this because that's not what I was invited to come talk to you about, but I just got inspired and motivated by the discussion you were having. So
we will return to the topic at the end of your talk, Tiago. Okay.
Anyway, so now I'll share my presentation. So you should be seeing it now. And yeah, so, oops, is it? Yeah. Okay, so I used this title, which was a bit different than the titles you had for the other lectures, because I like to play with words and the meaning of what we do. And what I'll try to talk to you about today is about some research we are doing in the lab that spans different aspects that I like to connect with what could happen in a kitchen, and eventually try to convince you that some of these things we're doing at a basic level may actually one day be relevant in the context of human health. And this is the program you are involved in. So I'm talking to you from Göttingen. Göttingen is this small university city in the middle of Germany, for those of you who may not know. The largest and biggest city here nearby is Hanover, so that's just on the trains, just half an hour away. Then we're also not too far from Frankfurt, an hour and 45, but Göttingen is really in the middle of Germany, and it's a small city with very good science. We have good research institutions around here, so it's good to be here and do science. And at least one of you is here in Göttingen, and I think she's enjoying, I hope. So your colleague Liana is working here with me. And Göttingen, just to show you a little bit more and to invite you to come and visit Göttingen. Göttingen is nice in the summer with a big main square where people gather and have a drink or a meal together. Well, of course, all of this before COVID, as you could imagine. But it's very nice. It has some important landmarks like this building down here. This was the observatory from which Gauss made some of his observations. He had his telescopes here, and he looked at the skies from this building. And he has this statue of this girl with the geese in the center of the city. So it's actually in this main square that you see here. And this is important because when PhD students finish their PhDs, then there's a ceremony and they have to climb this statue, and usually they try to climb as high as possible to leave a bouquet of flowers, and then they're supposed to kiss the girl. And there's a water fountain, as you can see here. So the trick is to try not to fall into the water and come back without any injuries. But sometimes there's colleagues that are so nice that they throw water balloons to the colleagues, and then they're all wet, and it's just fun to watch. So if you want to come and watch and throw some balloons at Liana, then she can try to to climb the statue as well. And I was looking at the EIT health website, and I was also inspired by some of the ideas. So I know there's a strong focus on this triangle, this knowledge triangle between research, education, which is what we're doing here and what you do in your PhDs, and then business. So there's a strong emphasis on this triangle, which I think makes sense. What I would like to bring in as well is that involving all of these, then we should have and consider science communication, which is important to really make the message of what we do more accessible to the community in general. And I think this is important for many reasons. Of course, we could spend time here talking about this. This is not the point. But I just wanted to bring this idea that I think keeping in mind and getting involved in science communication in general is something that I would also recommend that you all try to do, because this ultimately, I think we as scientists and as researchers owe the society in general, is that we communicate and explain what we do and why we do it, so that they also understand where their tax money is going, and not just that they see, oh, we're just funding scientists, but there's nothing coming out of what they do. So through science communication, you can convey all of this. And so I would like to invite you to engage in science communication activities, because this is important also for your future. Okay, now to the science, or to some of the ideas that underlie the science that we do. So for many years now, I've been interested in understanding what happens to proteins during this process that is represented here in simple ways that have to do with folding. So how do proteins acquire the three-dimensional structures that allow them to perform the functions they are supposed to perform in the cell, but also in a process that we call misfolding, which is an alternative pathway, if you will, where proteins may change their normal conformation into an abnormal conformation that we think is not so good and can cause disease. And this, we know, is associated with many different diseases, from cancer to viral infections to diabetes to neurodegeneration. And the problem is that protein folding in the cell is not a trivial event. And this is a schematic of a eukaryotic cell that you find in any textbook, and where you as EIT health students can easily recognize all the structures and organelles represented here in this schematic. But what these schematics fail to capture is that the intracellular environment is not really like it's shown here. So we don't have a lot of space in between organelles lying around. So what we have is a situation where the intracellular environment is very densely packed, where protein folding and the overall homeostasis is a challenge. And we know, for example, for proteins that enter the secretory pathway that goes through the ER, we know that there's a very high fraction of these proteins that has been estimated to be 70 or 80% that never becomes properly folded and that needs to be degraded. And for proteins that enter the ER, so the cell spends energy bringing the proteins in the ER, a lot of them never become functional, and the cell needs to send them back to the cytosol, use more energy to degrade the proteins in the proteasome and recycle the components, and then start over and make new polypeptide chains that hopefully will become a properly folded protein. So this is an expensive process, and it's expensive because, and it's worth it because the cell needs to really invest a lot in making sure that it doesn't make proteins that have problems and defects. So that's why these systems exist. And we've been interested in studying what are the consequences of protein misfolding and how cells find misfolded proteins and how they respond to the accumulation of these aggregated proteins. So it's like finding Wall-E. You know, you look at the one of these books and you look around and you need to find Wall-E in the crowd, and then eventually you find it. And this is what the cell needs to do, is to identify the protein that has problems, or the protein that is damaged and is not working properly anymore in a big crowd. So this is not so trivial. And to do this, then the cell has quality control mechanisms that involve a network of pathways and then individual proteins that contribute to this overall proteostasis. And here we can talk about the heat shock proteins or molecular chaperones, the degradation systems, the ubiquitin proteasome, autophagy. And then we talk about other pathways as well, signaling and response pathways that the cells use when they sense that there is a problem and that they need to adjust and produce more of some proteins than others to try to respond and compensate for any issues that might be occurring. So this is something that we now know is a complex network that we are trying to understand so that we know how we can regulate it when problems arise, but this is still something we need to study in a lot more detail. If we zoom in and focus on particular pathways, this becomes important because in the context of the diseases that I'll talk to you about, this main process of protein synthesis from the ribosome to folding and to degradation is important. So we have to consider basically these various steps. So the synthesis of the proteins, so the production, so this influences the levels of the proteins, but then the degradation by the proteasome or by autophagy, these also influence the levels of the proteins and of the reactions that may happen in between. So if we produce too much of a protein, this can be bad. If we degrade too little, it can be bad. Of course, if we degrade too much, it can also be bad. But this gives you an idea of how the overall homeostasis of proteins can be maintained. And then still there's a lot that can happen in terms of folding of the various proteins and how eventually this may lead to the accumulation of aggregated forms of some proteins. When this happens, if aggregated proteins accumulate, then we can have diseases like the ones I mentioned before, and among those we could have neurodegenerative diseases. And here I don't need to spend a lot of time in all of these. I mean, these are Alzheimer's, Parkinson's, ALS, Huntington's, all of these diseases shown here. We know that they affect particular areas in the brain more than others. And we also know that what they all have in common is the accumulation of aggregated proteins in the brain. So these little pictures you see here, they are the real protein inclusions that you find in the brains of patients that had these diseases after autopsy. So this is the ultimate way to really tell which disease someone had, especially if it's one of these neurodegenerative diseases. Now there's new tools and new approaches being developed to try to detect some of these protein aggregates while the person is still alive, but these are still not so useful, because there's also people that do not have disease that have apparently accumulation of aggregated proteins in the brain. So this brings us to other questions that we can come back later. But in any case, the idea is that people with these diseases accumulate aggregated proteins in the brain. And why does this happen? We are all different. We know this. Our genetics is different. But we know genetics explains only a minor fraction of people affected by these diseases, and probably not all cases, but fortunately some of them will start to go away. So the question is really, why do some of us end up developing some of these diseases and others don't? I mean, there's a lot of people living up to their 80s, 90s, and 100 years, and they don't seem to develop Alzheimer's. So why do some people develop Alzheimer's when they're 60 or 70? We don't know. So we need to study this. And of course, we know aging is the major risk factor, and that's why you are also involved in this PhD school to study aging and to study the consequences of healthy aging, or how we can promote healthy aging and not disease associated with the aging process. So I will now focus on Parkinson's disease. We study several of these diseases, or the molecular mechanisms associated with several of these diseases, but I'll focus on Parkinson's by using it just as an example. It's not that we don't care about the other diseases, but we need to study some of the proteins associated with the various diseases. And one of the proteins we study a lot is associated with Parkinson's. So for the remainder of the presentation, I'll just focus on Parkinson's. And for those of you who may not know, Parkinson's was described already more than 200 years ago by James Parkinson. And he observed some people that he saw walking in a park where he used to sit with this altered posture. So they had this imbalanced posture where they were curved forward. Some had some tremor. Their gait was also altered. They were moving with short steps. They had a lot of rigidity, and so this then enabled other neurologists to really define the key features, the key symptoms of Parkinson's disease, and they are the ones shown here. So this mnemonic trap helps you recognize the symptoms associated with Parkinson's. And then later on, what was discovered was that what happens in the brains of Parkinson's patients is that they lose neurons from a region called substantia nigra that you see here in a healthy brain, or from a healthy individual. You see this dark pigmented area that disappears in the brain of someone with Parkinson's, because those neurons that accumulate the pigment, the dark pigment, they die, and so you don't see this area in the brain of someone that had Parkinson's disease. And it's known now that the loss of those neurons is what causes the motor symptoms. Of course, one needs to keep in mind that Parkinson's is much more than just a movement disorder. So it's not just because of those neurons that we should study Parkinson's. There's many other neurons in the brain that may have problems as well. And that's important also for other features of the disease. But in any case, in the surviving neurons that you find in the nigra and also in other brain regions, you find those protein inclusions that we talked about. We think they are composed of amyloid fibrils, and these amyloid fibrils seem to be composed of alpha-synuclein, which is then a major component of these Lewy bodies. But alpha-synuclein is in Lewy bodies, not just in Parkinson's disease, but also in other diseases like dementia with Lewy bodies. And in another disease called multiple system atrophy, you find alpha-synuclein inclusions that are not Lewy bodies. Here, the inclusions are different, but you find them in oligodendroglial cells. So not in neurons, but in this type of glial cells in the brain. So together, we call these diseases synucleinopathies because they have the accumulation of alpha-synuclein. And our knowledge of Parkinson's disease evolved a lot since the identification of the first gene associated with Parkinson's, with familial forms of Parkinson's disease. And interestingly, it was at around the same time that the protein was identified in Lewy bodies that researchers identified a genetic association between the gene encoding for alpha-synuclein with familial forms of Parkinson's. And since then, many other genetic linkages have been established. The table is incomplete. I didn't put all the genes here and all the proteins here, because this list is still expanding. But this gives you an idea of the complexity of the problem. So there's multiple proteins. When we try to study the functions of these proteins, we get directed to different types of functions in the cell, suggesting that we need to really study cell physiology at different levels to understand how problems in different proteins that are involved in different pathways may contribute to the dysfunction that is characteristic in Parkinson's disease. In my lab, we've studied several of these proteins, and we are continuing to study some of them, but I will focus on alpha-synuclein for the remainder of the talk. And for those of you who may not be so familiar with alpha-synuclein, it is a relatively small protein of 140 amino acids. When we look at the amino acid content of the protein, we can define three main regions shown here in different colors. And then we know that the mutations associated with familial forms seem to be in this N-terminal region. So all of these shown here in black, A30P, 46K, etc., they are mutations associated with familial forms of the disease. And then on top, you see these letters. These are post-translational modifications that have been described in alpha-synuclein, and we'll come back to them in a little bit. Now, we know the protein is abundant in the brain, but we also know it's abundant, for example, in the blood, and it exists in other tissues as well, including the heart. But no one really studies alpha-synuclein in those tissues, which is a shame, because maybe it's doing something relevant there as well, and we still have only very poor information about what it does outside the brain, and it could be very relevant also to explain some of the features that are not just related to the accumulation of alpha-synuclein in the brain. So maybe some of you can study this in the future. Now, what's interesting is that the protein was initially described in '88 as a neuron-specific protein that was localized in the nucleus and in the presynaptic nerve terminal. So these were old studies when antibodies for alpha-synuclein became available, and where people were doing stainings, and they found it in the nucleus and then also in the presynaptic terminal. But then for a long time, no one really cared about synuclein in the nucleus, because they thought, oh, that was just an artifact of the antibody, and the protein is really just presynaptic, so it must be doing something in the presynaptic compartment. And this doubt remained, and to be honest, it still remains. There's still some people that don't really care too much about what it does in the nucleus, or don't even believe it may exist in the nucleus. But we looked into this recently, and we published some studies where we reported some of the effects of alpha-synuclein in the nucleus. So we found in not only in cultured cell lines, which of course are not as physiological as neuronal cultures or brains from animals, but we found synuclein in the nucleus. We found evidence for enhanced phosphorylation on serine 129 in the nuclear alpha-synuclein. We found that alpha-synuclein expression in the nucleus can inhibit histone acetylation. So looking here at acetylation of histone H3, we found that in animals expressing wild type or mutant alpha-synuclein, the levels of acetylation were reduced. By doing gene expression analysis, we found several genes whose expression was altered when you overexpress alpha-synuclein. And then we found also that maybe nuclear alpha-synuclein reduces cytotoxicity. So maybe it's trying to do something in the nucleus that reduces cytotoxicity. And this is something we're still studying. But these studies were done in models and in cells and in animals. So the question is there really alpha-synuclein in the human brain? And if there is, then is there any disease-specific difference in the content of nuclear alpha-synuclein? And what are the potential consequences of altered levels of synuclein in the nucleus of cells in the brain? So these sections I show you here These are from human brains, so post-mortem, of course, from either a dementia with Lewy body case. So these are human brain sections stained for the nuclei in blue, and with antibodies for alpha-synuclein in green. So here, this is a general alpha-synuclein antibody, and here on the right, it's a phosphorylation-specific antibody. And you see on top the DLB case, and on the bottom, a control case, so someone that did not have any evidence for a Lewy body disease. And what was interesting to notice was that we found evidence for some alpha-synuclein in the nucleus. So here you see some signal overlapping with the nuclear staining, and I'll show you more evidence we obtained by doing fractionation of the tissue homogenate and separating nuclei from the supernatants from the rest of the material. And here, if we just look at the nuclei, so these are nuclei stained for alpha-synuclein, we see some staining in the nuclei, so confirming the previous findings. But also on Western blot, we could detect always some synuclein in the nuclear fraction. So of course, there's more in the cytosol, but in the nuclear fractions, here labeled with N, there's always some alpha-synuclein as well. These are just the controls, the cytosolic control and the nuclear loading control. And here, another control we used, this was tissue from an alpha-synuclein knockout mouse to show that there's really no signal when you probe with the synuclein antibody. So the signal that you see there is specific and suggests that there is some alpha-synuclein in the nucleus. Now, what was interesting was that we did not find evidence of altered levels of alpha-synuclein in the nucleus in samples from patients with disease versus controls. We found perhaps evidence of some oligomeric forms of alpha-synuclein that ran a little higher than the monomeric form. So this is something that we are looking into. But what was interesting was to see that there were other differences that were related to phosphorylation. So when we used an antibody specific for phosphorylation on serine 129, and when we quantified several samples we looked at, we found that there was more phosphorylated synuclein in the nucleus in tissue from DLB patients than in control. So this is something we are now following up and studying in greater detail. So just to summarize this first part, I showed you that alpha-synuclein can indeed be present in the nucleus, that the levels of phosphorylation seem to be different in normal physiology. So in someone that was aging normally, if you will, and in someone with disease, it seems that there's more phosphorylated alpha-synuclein and perhaps more oligomers of the protein as well. So we are now looking into the possible effects of these phosphorylated forms of alpha-synuclein in the nucleus, and maybe in the future we'll have some more information. But I would like to go back to this general question: why does this protein become toxic, and how does it cause synucleinopathies? And here I put cause in italics because we are still assuming it causes synucleinopathies. We cannot say for sure that the aggregation of alpha-synuclein or a beta or tau in Alzheimer's disease cause the disease. We know they are there, but we cannot say that they actually cause disease. So when we think about the proteins, this is for alpha-synuclein, this is the structure that people have determined for alpha-synuclein in the context of some membrane interaction. So it forms some alpha helices, and it's thought that it's through these alpha helices that it interacts with membranes. And then it has this C-terminal region that is disordered. So there's not a lot of structure in this region, which allows the protein to interact with a lot of its interacting partners. But we don't know much about it. But this is the structure that was determined based on recombinant synuclein purified in vitro. And the issue is that in the context of a normal cell, you don't have the protein alone without its interacting partners or without membranes or without lipids or other molecules. So you have to think of a situation where it's not just the primary amino acid sequence that is encoded by the gene that matters. So other things become important in the context of real biology. And these can be, for example, protein post-translation modifications, which are determinants of protein folding, localization, and function. And these remind me of this cathedral in Barcelona, which I'm sure a lot of you have seen. This cathedral has been under construction for more than 150 years, and it's still not finished. So every time I go there, every couple of years, I see that there's some new painting or some new structure added. And this is like what happens to proteins. From the moment they are produced from the ribosome, their journey really starts. And so you have any protein, alpha-synuclein, or any of your favorite proteins, during the normal lifetime of these proteins, they become modified by different post-translation modifications. And in some regions, it could be that only one type of modification can take place and not another. In another region, you could have multiple types of modifications happen, depending on which one the cell decides to add. But you can have a situation where the complete set of modifications may lead to a conformational change that, in the case of some proteins, can be detrimental and can trigger aggregation. So I think we need to really understand how these modifications affect the folding and function of the proteins, and also the localization, because as I showed you before, maybe phosphorylation on serine 129, for example, increases the localization of synuclein in the nucleus. So maybe there's other modifications that affect its distribution in the cell as well. And these modifications are not there just by chance. They were evolved by nature over millions of years of evolution to increase the complexity of proteomes. So you know probably that in our human genome, we have about 20 to 25,000 genes. Then through alternative splicing, we generate about 100,000 transcripts, but it's through post-translation modifications that we expand this complexity of the proteome by producing what is estimated to be more than 1 million different types of proteins. So they are really important, and they are important also in the context of intrinsically disordered proteins, which are proteins that are either entirely disordered or that have regions that are disordered. And I showed you that alpha-synuclein in many conditions is completely disordered, and in other conditions has a highly disordered C-terminal region, which can be subject to different modifications. So they are like condiments that the cell uses to give flavor or different flavors to the protein depending on what they need to do. So we started looking at this as well already several years ago, and we've been studying not only phosphorylation, which we know is relevant in the context of Parkinson's, but also other modifications. We asked whether specific modifications or sets of modifications affect the aggregation and behavior and toxicity of alpha-synuclein. And we noticed years ago that there is a connection between diabetes and Parkinson's disease, and we don't know very well why this is the case, but it seems that patients with diabetes have increased risk for developing Parkinson's disease. And we hypothesized that maybe this could have something to do with glycation. Glycation is a type of post-translation modification that is non-enzymatic, and it consists of the reaction of proteins with sugars. So when certain amino acids that are more reactive interact with sugars like glucose or other sugars, a series of reactions can take place, and eventually you can have the formation of advanced glycation end products, which are typically irreversible, and they lead to the cross-linking of peptides and proteins that, of course, changes their function and and their ability to do what they need to do in the cell. So using mass spectrometry analysis, we extracted alpha-synuclein from cells and from mouse brain tissue and eventually from human brain tissue. And we looked at the protein and we found there were several lysine residues, primarily in the N-terminal region of alpha-synuclein, that were glycated. And they're shown here in red, these various lysine residues. And if I show you the whole sequence of alpha-synuclein, you see that the protein has 15 lysine residues that span almost the whole protein. But the ones that we found more consistently glycated were the ones shown in red, so the ones closer to the N-terminal region, which is an important region for interacting with membranes, as I mentioned earlier. So we thought that maybe this could do something relevant and important in the context of the behavior of the protein. And we started asking a bunch of questions, whether glycation had a physiological role or a pathological role, what was the most toxic form of the protein, in which conditions alpha-synuclein could be glycated, whether this was a cause or a consequence of glycation. So we asked all these questions and we tried to address them using models. And of course, we would like to work with these types of models, but this is not possible. So we have to be creative and go to the models we are allowed to work with. And we really go and explore a variety of models. So we use very simple eukaryotic models like yeast cells, and we use also more complex models like the ones Liana is also using, mouse models and rat models. But we use Drosophila whenever the questions we need to ask suggest that maybe Drosophila is a good model, or C. elegans, or primary cultures of neurons and glia. So we use all of these models, but keeping in mind that they are just models. And so this is, I think, something that sometimes we tend to forget. But these are just models, and none of these models completely recapitulates what happens in a human disease, because they are not humans. But sometimes people get very attached to mouse models. Oh, they are mammalian systems, and therefore they are much better than fly. And that's true for many questions, but it's not true for all questions. There are many instances where simple models can be more valuable because the genetic tricks you can do with these different types of models may allow you to address certain questions in a more direct way. So keep an open mind to all the possible models, keeping also in mind that they are models and eventually they're all wrong, but some are useful. And this is how we think about the research we do. So because we know they're all wrong, we combine and we use a lot of different models. And here I show you some experiments we did with patient-derived iPS cells. So we had cells from a control individual and cells from an individual carrying a triplication of the alpha-synuclein gene. And these patients, or these people that carry triplication of the alpha-synuclein gene, they develop Parkinson's disease. If they live long enough, they will develop Parkinson's disease, and we know that they express more alpha-synuclein. So we took those same cells and we knocked down alpha-synuclein using RNAi to kind of correct the levels of expression back to normal levels. And then we incubated these cells with methyl glyoxal, this small aldehyde shown here, with different concentrations. And we found that the cells with the higher levels of alpha-synuclein, with 3 copies of the synuclein gene, were more sensitive to the toxicity of methyl glyoxal than the control cells or the cells that were corrected for the levels of synuclein. So there was an association between the levels of synuclein and the toxicity of methyl glyoxal. And the next thing we did was to go to mouse models, and we took either control mice, so wild type mice, or transgenic mice expressing human alpha-synuclein, and we injected in one side of the brain just PBS, and in the other side of the brain methyl glyoxal. So and then we stained the substantia nigra for tyrosine hydroxylase, which is a marker of dopaminergic neurons. We stained for nuclei in blue, and we stained for carboxyethyl lysine, which is one type of glycation induced by methylglyoxal that we can detect using an antibody. And what was interesting was to see that, of course, in the control situation where the side of the brain was injected with PBS, we didn't see a big difference in terms of the number of Th positive neurons in the nigra. But on the other hand, when we injected methylglyoxal, we saw a lot less Th positive neurons in the transgenic animals that expressed more alpha-synuclein than in the wild type animals. And we also saw a lot more protein glycation in those animals. So again, this suggests, it doesn't prove, but it suggests that if you have more alpha-synuclein together with glycation, this is a bad combination. This can do something bad for the animals. The next thing we did was to ask our friend and collaborator, Flav Giorgini in Leicester, to test some of his flies that express human alpha-synuclein, so they're transgenic for human alpha-synuclein, to test whether glycation would affect their motor performance or their longevity. And in flies, as some of you might know, you can assess motor performance by doing a simple test that involves measuring the time they take to climb up the wall of a glass tube where you can keep them. And here there's a lot of conditions, so a lot of controls, but I'll ask you to focus just on the purple and the red bar. So we looked at the flies at different time points, day 10, 20, or 30, and we compared these flies. The purple bar, for our purpose here, are the control flies, and then the red bar are flies that have increased glycation. And the way to do this is not by injecting artificial amounts of the glycating agent in the flies. What we did here was to use fly genetics. So we knocked down, using RNAi, an enzyme called glyoxylase 1, and glyoxylase 1 is important in methylglyoxal homeostasis because it converts methylglyoxal to lactate to detoxify and to avoid the accumulation of this toxic and reactive molecule. And basically what you see is that if you knock down this enzyme, then the motor performance of the flies is always worse than the control because they have more methylglyoxal, they have synuclein, and again, they have more problems. It doesn't prove that there is a direct effect, but it again supports this idea that if you have more synuclein and more glycation, this is bad. And then, of course, we did a lot of studies in vitro and in cells to try to understand what was going on, and using simple assays like an aggregation assay, using size exclusion chromatography and electron microscopy, we saw that the treatment with methylglyoxal was actually promoting the formation of oligomers, not necessarily the mature fibrils that we see clearly here under the electron microscope, or that we can detect using thioflavin T, because they bind more of this dye called thioflavin T. But we could see the accumulation of oligomeric species of the protein. And as some of you might know, we believe that maybe these oligomers are more toxic than the mature fibrils. So this was very interesting. And then we did other experiments. So Hugo Miranda, a former postdoc in my lab that is now independent, he did some of these experiments where in cells expressing alpha-synuclein, we blocked protein translation with cyclohexamide, and we treated them with either vehicle or methylglyoxal. And then we looked at the half-life of the protein, and we saw that with methylglyoxal, the half-life of the protein was increased, suggesting that the protein was not being degraded as quickly. And so we were in the situation of proteostasis imbalance. And of course, what we want to promote healthy aging is to tip the proteostasis balance to the other side. And we looked and we found some molecules that were already described in the literature that act as methylglyoxal scavengers, and two of them are shown here. And we fed the flies with some of these compounds with increasing concentrations, and then asked, do the flies improve their motor performance? And indeed, this was surprising, but we saw some improvement. I mean, it was not a complete rescue by any means, but the motor performance seemed to improve with increasing concentration of these molecules, suggesting that maybe if we reduce glycation, this then is beneficial for the flies. Now in the lab, we're studying not just glycation by methyl glyoxal, but by a variety of other sugars that we think could be relevant also in the context of diabetes and other human conditions that one could consider. And using, again, mass spectrometry analysis, we can detect that different sugars can glycate alpha-synuclein in different lysine residues. So we are studying this in great detail. And interestingly, different sugars lead to the formation of different types of alpha-synuclein aggregates. So this makes the whole study very complicated, because how do we know what is the right balance of sugars that one should have to avoid the formation of toxic species of the protein? This will be really, really difficult to achieve, but we can really see that while some sugars reduce the formation of fibrillar forms, as you see here, more oligomeric forms, then other sugars still enable the formation of fibrillar forms with higher thioflavin T binding and with clear fibrils being detected. And we can detect the fibrils and the aggregates using different assays. I will not go into detail there. So I'll just show you a quick summary, a graphical summary of what we did. We did experiments in mice, in flies, in various types of cells, where we could show that increased levels of synuclein together with glycation were bad. We found that perhaps glycation is promoting the accumulation of toxic species that are like these oligomers. And then we did experiments in other systems. I didn't show you all of these, in yeast and other simple cell systems, to really identify what was happening to the protein. And it seems that the protein is not being degraded properly. And we showed also that methyl glyoxal scavengers might be protective against the toxicity associated with glycation. So does this make sense? Or is this just our imagination or interpretation of the data? And what I'd like to try to convince you of is that this seems to make sense also if you look at the genetics of Parkinson's disease, because there's a gene called PARK7 that encodes for a protein that is known as DJ1, that and mutations in this gene are associated with autosomal recessive forms of Parkinson's disease. So these are loss of function mutations that seem to reduce the function of this protein. And for a long time, this protein was thought to be involved just in oxidative stress responses and in protecting against oxidative stress. But there were some more recent studies that showed that this protein seems to have glyoxylase function and also deglycase activity. So we are now collaborating with some of these colleagues in France that reported deglycase activity in DJ1 to see if it can actually deglycate alpha-synuclein, which should be really important. And just by chance, we were studying DJ1 also on the side and how it could affect alpha-synuclein. And some years ago, we found that it interacts with synuclein and it attenuates the aggregation and toxicity of alpha-synuclein also in laboratory models. In human cells expressing the two proteins, we could see a reduction in toxicity. And here, we weren't even thinking about glycation or that DJ1 might be reducing alpha-synuclein glycation. And we saw evidence that there was a beneficial effect of the co-expression of the functional form of that protein. So what is the message of this part? The message is not that glycation causes Parkinson's disease immediately, because not all patients with diabetes will get Parkinson's. But maybe glycation can be one of these insults that can tip the proteostasis balance towards the wrong side if then you have some SNP that leads to the increased production of alpha-synuclein, or some other problem that may lead to the accumulation of alpha-synuclein. Then you could be in this situation where combining genetics and environmental risk factors could lead to the development of a disease like Parkinson's. And we can come back to this at the end if you have questions, because there are some good questions to be asked regarding glycation. Now, I would like to just end by highlighting some recent methods and techniques that I think are very cool that we are using to study protein aggregation. And this is a simple schematic of the process of protein aggregation, where you start with monomeric proteins and you end up with big fibrils and aggregates that we can see. But the question is really, how do we find them in a cell? If we want to study all these different species, the big aggregates maybe we can find with antibodies. This has been done for a long time. But how do we find Wally? And for this, we think we need to use super resolution microscopy, or what some people call nanoscopy, because now you're not in the micro scale, you are in the nanoscale. And we all know size matters. There's different tools you use to look at objects of different sizes. So you use telescopes to look at stars and planets and galaxies. And then we need to use microscopes to look at cells and certain subcellular compartments. But when you want to study proteins, then you need to really go to a scale that is not so easy to achieve with a typical light microscope. So you need to use nanoscopy and electron microscopy, and now cryo electron microscopy that is so popular these days. So we cannot do it with light microscopy because of the limit of resolution that we know that microscopes have, light microscopes. And this limit of resolution is around 250 nanometers. So this means that if two objects are closer together than 250 nanometers, you will see them as a single object. And this is a problem, and we want to study these tiny little inclusions. So a colleague here in Göttingen, Stefan Hell, he's a physicist, and he was very clever because he came up with a way to extend this Ernst-Abbe equation that you see here in Jena, in a university in Germany, that tells you that the resolution is proportional to the wavelength and inversely proportional to the numerical aperture of the objective. And basically he extended the equation and added some other parameters, and he was able to break this resolution limit. So now, instead of having this type of resolution with a typical confocal microscope, you can have much greater resolution of structures inside the cell using, for example, this STED microscopy. There's different types of super resolution microscopy. This is just one of them that is very popular here in Göttingen, and we are using to try to image protein inclusions. And I'll just show you that this actually works. So here we have human cells expressing different variants of alpha-synuclein, either wild type protein or other proteins that form smaller inclusions or even smaller oligomers. And using STED, we can actually start to resolve some of these structures. And we hope that we may learn about the architecture of the proteins in these types of inclusions using these more advanced types of microscopy. So I will stop here, and I'll just tell you where we stand these days. So in these neurodegenerative diseases, clinicians are good at identifying the features. They can detect the clinical symptoms, and then we have, for some of these diseases, we have therapies that treat some of the symptoms, and Parkinson is probably the best example, where some therapies treat the motor symptoms, but then we don't know the causes in great detail. We have a lot of ideas, but we don't really understand the causes of all these diseases, and this is where we need to invest a lot so that we have future therapies for all of these problems. So to end, just to show you the current group that works here with me, these are the students. Liana is there as well. And I have to thank, of course, a lot of collaborators with whom we do a lot of this exciting work, and the funding agencies. And I thank you for your attention as well. So I'll be happy to take questions and discuss some of these ideas with you. Thank you.
Thank you, Tiago. Thank you for this really very interesting talk. And so I open this talk for discussion. So students, if you want to raise your questions. So I see Alejandro raising his hand. Alejandro, please.
Hi, Tiago. Thank you for your presentation. I really liked it. And I'm not into the Parkinson's field or the alpha-synuclein, but I have a question about the glycation and the association that it has to diabetes and high blood glucose. So I know this happens like with many proteins like hemoglobin, and it's also like a biomarker for Parkinson's. I mean for diabetes.
Yeah.
And is there a way that low blood glucose reverses this glycation? Because I see that high blood glucose like possibly glycates, but I don't know if the other way around happens.
Yeah, that's a very good point. So glycation is actually, as you mentioned, something that we use to diagnose diabetes. And it's a modification that happens not just in alpha-synuclein, but in many other proteins as well. So we don't know exactly how we can reduce glycation, because in many instances it seems to be irreversible. But now that there are some enzymes that people describe as having deglycase activity or glyoxylase activity, maybe there are ways that we didn't know before that can be used to reduce protein glycation, whether it's of hemoglobin or other proteins. This remains to be determined. But it's not just glucose that glycates hemoglobin and other proteins. There's many sugars and molecules that have reactive groups that would eventually react with proteins just as glucose would, that lead to the modification of proteins that could cause them to aggregate and accumulate in these forms that are probably toxic to cells. So it's a complex question. There is evidence from studies in Parkinson's patients using anti-diabetic drugs, suggesting that indeed they have benefit in Parkinson's patients as well. And the molecular mechanisms are not well understood. So it could be that it is through effects on glycation, but we don't know that in detail yet. So there's still a lot to learn.
Thank you, Alejandro. Thank you, Tiago. I also take the opportunity to ask people following the talk by at YouTube, if you have questions, please leave your questions in the chat, then I can just read for Tiago. Other questions from our students? No questions? Too shy today.
Or we can go back to the topics in the beginning.
No, but Tiago, actually, let me just raise one issue, which is indirectly related with your talk and the glycation of alpha-synuclein, but also other proteins. So according to the data that you have and the data from your research and data published in the literature, so for what would be the consequences of these data for, I would say, human diet in terms of intake of different sugars? And so, but not only related with actually concentration of sugars in the body, but mainly due to the timing of intake of different sugars. So whether or not, I would say, Western diets and I would say intake of high concentrations of sugars, but I would say in a sustainable way versus fasting, I would say fasting periods of fasting would play a role, whether in say in glycation, in pathological let's sort of pathological glycation of proteins that can lead to neurodegeneration. Do you see any link there?
Mm-hmm. Yeah, that's an excellent point. We are interested in looking at these peaks of high sugar and low sugar, because this is what happens during our normal diets, right? We eat or we eat something with sugar, and sugar levels go up and then they go down. So we think this is really relevant for determining the effects on some of these proteins. And of course, then we need to consider that there might be response or defense mechanisms to deal with the glycation. There's also, of course, proteins that even if they are glycated, they can be degraded. So the half-lives of the protein are very important. Some proteins that are short-lived, it won't matter so much if they are glycated, because maybe they won't be as glycated and they can be degraded and never cause problems. And other proteins that are long-lived, if they are glycated, their half-life could become even longer, and then they could accumulate long enough that they could cause problems. And for alpha-synuclein, this is very relevant because alpha-synuclein is a very long-lived protein in the brain. So if you compare the half-life of alpha-synuclein with the half-life of other synaptic proteins, alpha-synuclein has a much longer half-life than most of those other proteins. So if it then faces cycles of exposure to high levels of sugars and cycles of exposure to lower levels and then again higher, this can really affect the biology of the protein. And we are trying to study this as well. That's something we want to study. One difficulty is that we don't have necessarily good tools to study synuclein glycation. Until now, there was no antibody for glycated alpha-synuclein. We think we have some now that we developed. We're still testing them, but we now think we have antibodies that we can use to go and take human brain tissue and look to see if there is glycated synuclein in human brain tissue. So hopefully we'll have more news for future talks.
Thank you, Tiago.
So there's a question here in the chat by Isabel Rocha. Isabel, it's nice to see you're there. It's been a long time since we saw each other. So let's see the question. How do I envisage the translation to the full living body, normal and sick, of the cellular and molecular research? What are the benefits and pitfalls of the cell and molecular research in this scope translation? Yeah, so absolutely. So this is research we're doing at the molecular level, and we think that we need to understand what happens at the molecular level, because eventually this is how things will go to the tissue level and eventually to the organ level and ultimately to the whole body level. So here, if we talk about synuclein in particular, this is relevant because synuclein does what it's supposed to do in the presynaptic compartment or in the nucleus. But now we know that not just in Parkinson's, but in many of these neurodegenerative diseases, there seems to be spreading of pathology, meaning that the proteins can accumulate in some parts of the brain or even outside of the brain. And then through a mechanism that is still not very well understood, but everyone refers to as a prion-like mechanism, maybe the pathology spreads from one cell to the next, and from one brain region to the next, and from one organ to the next. And so eventually, because you have glycated protein, or if you have glycated protein accumulating in the brain, or even in the gut where sugars enter the bloodstream, if you start to have glycated proteins there, maybe this triggers the aggregation of the protein, and this would then trigger with time, with aging, the spreading of pathology that eventually could come to the brain. And so, of course, we don't know exactly how to stop this process. The studies we're doing are still in simple models, but eventually the goal is that this research can be translated into an application that would affect the individual everywhere where the problems might be taking place. So yeah, we hope to translate some of these findings eventually. And I don't know if I answered the question completely, but this is my view of the question that Isabel asked.
Thank you, Isabel. Thank you, Tiago. So more questions? We still have some time. If you have questions, Tiago, please.
Professor, this glycan, they interact with some proteins like Siglec class of proteins, and they are related with neuroinflammation. This is a way to.
Mm-hmm. Yeah, I'm not sure I understood the question completely, but it's true. So I didn't cover all of this because there'd be no time. But glycated proteins and these, especially these advanced glycation end products, they are known to activate receptors like the RAGE receptor, the receptor for advanced glycation end products that exists in certain glial cells, for example. And so cells or some types of cells can sense glycated proteins and then elicit an inflammatory response to signal that there's something bad accumulating in this particular tissue or region of the organ. So yeah, the complexity is much greater than what I was able to present here in such a short time. But of course, these glycated products can trigger inflammatory responses that, if then they are sustained over time, then they can lead to, of course, detrimental problems, because too much neuroinflammation or inflammation in general is not good. So this is a consequence of some of these processes that are taking place, for sure.
Thank you.
And thank you, Tiago. So more questions before closing the session?
I have one question.
Yes, Masa, please.
I was wondering if glycation and phosphorylation are also important for like other diseases which you mentioned, where alpha-synuclein is also involved, because I'm not really in the field of neurology, so it's interesting for me.
Yeah, no, very good question. So no one asked this, but this is an important question. Why would glycation be important just in the context of alpha-synuclein? And of course, that's not the case. So we heard from Alejandro that hemoglobin can be glycated in diabetes, and we use that as a marker. And we know that many other proteins can be glycated in different tissues. So here we were specifically looking at alpha-synuclein and glycation. So fortunately, we found something. But we've done other studies with other proteins like huntingtin, which is associated with Huntington's disease. And we also found that if we increase glycation of huntingtin protein, this is also detrimental. So I'm sure that there are more examples in other diseases where glycation would also have detrimental effects on other proteins. The issue that we need to always keep in mind is that it's not always possible to predict whether glycation will cause problems in other proteins, because then this has to do with the way cells handle those other proteins. So if the half-life of the protein is shorter, then the problem will not be as bad. If the cell has very good ways for keeping the proteins folded through chaperones or interactions with other cellular compartments, maybe this will not be such a big problem, because the protein will always be doing something and glycation may not affect the function as much. So it always depends on the protein, but we know that glycation and phosphorylation, like you mentioned, in the context of other proteins can be detrimental as well. For example, in Alzheimer's, hyperphosphorylation of tau leads to its accumulation in neurofibrillary tangles. And so we had many examples where altered post-translation modifications can indeed affect the function of the proteins and could be associated with disease. So that's known for sure.
So in that case, that means that the best that we can have is to have like the best balance between the normal glycation and the pathological glycation, right? So what do you think that, or how do you envision that this can be improved or achieved?
Yeah, so ideally, we all like to eat chocolate and eat candy and drink Coke. I mean, I do. Ideally, what we would want is to be able to eat a lot and do all the bad things we do, and then take a magic pill that would come and resolve all these bad things that we do. So until this happens, we need to, of course, balance our diet, be careful with the exercise we do, with the sleeping habits, with the cognitive habits we have. Now, this is what we all know from studies in many different diseases, but these things we know also help in the context of neurodegenerative diseases. So we should be careful with our diet for sure, to try to minimize the potential problems that would come from imbalances in glycation. And it's not always easy to control just by changing our diet, because there could be genetic alterations or genetic factors that also change the way we metabolize sugars, and then those we could not control. But at least the ones we can control, maybe we should try to control, so that we minimize the risk for developing disease. That's how I try to do it sometimes.
Thank you, Carlos. We have a follow-up question from Professor Isabel Rocha. So, Tiago, following a bit this last question, so the last one, do you think that if we be able to tackle a cluster of dysfunctional brain cells, we will be able in the future to deal or treat the various phenotypic, for instance, diseases related with their own dysfunction, meaning that the future of therapeutics is in the brain clusters of cells? Would you like to comment on that from you? And actually, this question aligns very well with the yesterday keynote lecture from Thomas von Zielinski about senescent cells. And Tiago, do you have a comment on this?
Yeah, I think this is an interesting thought. And if we are able to pinpoint where dysfunction starts, which are the cells that start to be dysfunctional first, maybe we can stop the problem there and then prevent additional problems from happening in other more distal areas of the organ or the whole body. So I think that's an interesting concept. I think this requires also a much greater understanding, not just of the disease mechanisms, but also of the signaling and the symptoms, so that we can have better biomarkers. So we need to be able to diagnose these diseases much earlier to know, okay, now is the time to intervene, because if we intervene only in 10 years, when the phenotypes are all overt, then it might be too late. So we need to improve diagnostics as well. But it's possible that if we identify the problems early enough, when they are circumscribed to a specific cluster of cells, maybe that's when we can still do something about it. So I think that would be the way to go, to try to go as early as possible when the issues are still concentrated in a particular cell.
Thank you, Tiago. Thank you, Isabel. More questions? So it seems that we don't have more questions. No, Isabel is typing also just to share what you said before, exercise, etc., goes in line with what we are seeing in patients with reflex syncope and atrial fibrillation. The usage of no pharmacological measures to reverse disease phenotypes. Yes. Many thanks for you, Tiago. Many thanks all our students for the discussion. Well, taking actually the note from yesterday from Thomas von Zielinski, by attending a lecture such as this lecture, we extend our healthy life extra 15 minutes due to the cognitive stimulation and the benefits. So 15 minutes from yesterday, 15 minutes today. So we have more 30 minutes of healthy life expectancy. Thank you so much, Tiago. Thank you so much for all the students. I'm now going just to stop the session on YouTube. Many thanks for those that have been following on YouTube and that can follow this lecture later. So goodbye.
Thank you. Bye-bye.
Thank you. Let me just, okay, it's now close the YouTube session. So Tiago, many thanks. See you around. So we will keep inviting you for different, challenges.
Yeah.
And many thanks for your involvement.
No,
so,
thanks for the invitation, Ron. It was a pleasure.
Thank you, Tiago.
Good luck, everyone.
So to the students, Liana, so we have the alumni session, the ice-breaking session. So how should we proceed? Should I leave this platform open? Liana, you just wrote that you have a different link to the alumni session. Yes.
Yes, I created a EIT Health alumni link, and I already sent to everyone. I hope you all got it, both on WhatsApp and on your emails. And we will do it over there because it's easier for the breakout rooms and everything, so we don't bother you. And we also want to stay for the lunch time.
Okay, together. Liana. So, everybody, thank you so much for your initiative and for keep being involved. We will return to this same room at 2 o'clock, so 3 Central Europe time, correct? Fine. Thank you. Bye-bye.